Rodney, and all of our amazing presenters from part one. They left very big shoes to fill. We're really excited to share with you part two of today's workshop, Building a Foundation for Success in Clinical Research. So now you have a foundational understanding of science and data sets, preclinical models, and translational research. We want to cover what it takes to move into clinical development and how patients can really be the drivers of this important process. What does it mean to be a good partner? How can we, as advocates or advocacy organizations, drive progress? I'm excited to be joined in this session by Vanessa Vogel Farley of RareX and the Duke 15Q Alliance, and by Dr. Michael Grosso of Daiichi Sankyo. I thought that I might start by showing and sharing what the TSC Alliance has done to really build collaborative resources and tools to achieve and increase the interest in TSC drug development. Again, I want to emphasize we built our research platform over 35 years. And if we can just advance these slides to our research platform. You can see on the left that we have some tools that fit more into the previous conversation and are directed more towards hypothesis generation through research grants and drug testing in preclinical models, which are all meant to de-risk drug development in TSC. And on the right side of this graphic is where we're going to focus today and how we as advocates working in partnership with academia and industry can share our real world experiences on ad boards, through natural history databases or patient registries, by truly understanding the impact on quality of life through the collection of patient-reported outcomes. And in the case of the TSC Alliance, that's linked to our natural history database along with biosamples and how we can work towards building centers of excellence to drive clinical trial infrastructure, such as we have with our Clinical Research Consortium. All the tools at the TSC Alliance were built to push drug development from the left and pull it from the right. They're resources accessible to investigators, whether academia or industry, and definitely in partnership with our patient community. This is just one example of a research platform. We took time to build this by adding each element, as you can see, because the year that we put them into our research platform is listed, and in a way that we could sustain our program as a whole. It's scalable, meaning we can add more biosamples or more participants to our natural history database in a year where fundraising is going very well, or we can scale back in challenging times like during COVID-19. We built our program to be particularly flexible for that reason. The model I'm sharing talks about clinical trials and real-world experience. There are phases to clinical trial and development, and we'll include those in the overview of clinical terms in the resources that you receive after this workshop. But I want to start today's session by introducing our first speaker, Vanessa Vogel Farley. Vanessa is the Research and Data Governance Platform Lead for RareX and also serves as the Executive Director of Duke 15Q Alliance. Previously, she worked at the University of Minnesota Center for Neurobehavioral Development and as the Clinical Research Coordinator for the Division of Developmental Medicine Laboratory of Cognitive Neuroscience at Boston Children's Hospital, collaborating with researchers at MIT and Harvard. She has over 15 years experience in data collection, nonprofit strategy, patient advocacy, and beyond. And I can tell you that this woman is a powerhouse. So Vanessa, I'm going to turn it over to you.
Thank you, Kari. I'm going to share my screen. And I hope you can see the proper screen. Give me a thumbs up if you can. Great. So as Kari introduced me in my background, I have now over 20 years of experience in collecting data, which is kind of crazy how quickly that adds up. And I am really proud as a person who's collected data over time, my 15 years of experiential data collection in the laboratories of cognitive neuroscience studying child development in that space, I really came to learn a lot about data collection, especially in rare disease populations, and how we needed to approach it maybe a little bit differently. And then during my 5 years of being the executive director of the Do15Q Alliance, really being able to take some of that knowledge and those shared experiences and apply it to how we collect data at Do15Q and in collaboration with Angelman syndrome. But today, I think what I really want to bring forward is the endeavor that is called RareX. And I serve as their research and data governance lead. And what the purpose of RareX is to really help bring forward some infrastructure and some shared learnings where oftentimes as rare disease patient groups, we're talked about, we stand in all of these places and say, we need to collect data. That's what we're told. You know, Dr. Woodcock from FDA, Dr. Mullins from FDA, CDC, all of the people go, the only case the best case scenario is collecting data to get your natural history study, that's the only way we're going to get to the end of towards therapeutics. And oftentimes they get off the stage and they don't tell you how. And what ends up coming that these patient groups and all of us patient groups end up having to become experts in all of the things that go into data collection, whether it be the infrastructure for governance, the infrastructure for actually the tech side of things, the infrastructure for data sharing, and all of these other areas where at the end of the day, a patient group, it's really, really tough to, as you're kind of bring your community together, as you're trying to figure out what your education looks like, they're trying to support all these families who are being newly diagnosed. This is an entire other realm of things that we need better support in making this a shared environment. So at the core of this, what RareX teaches to patients and its importance to progress is to accelerate disease understanding and the production and approval of therapies. First, by characterizing your disease. We talk about natural history studies. Kari talked a little bit about the Tuberous Sclerosis Alliance's pipeline where they have the natural history data. They have their real world evidence part. But how do we characterize the diagnosis, the clinical manifestations, the change over time, and all of those things? Oftentimes we talk about natural history studies. Everybody hears about those, and those are really, really paramount to collect. How does the disease present, and how does it change over time? Then figuring out and categorizing and organizing your data that's out there. Are there similar diseases to collect data in the space that you can really learn from? Oftentimes you feel like you're in your own bubble because we are all rare. But there's a lot more in common, especially when it comes to data collection and towards clinical trial development that we need to share. And so really going into that categorization and organization, saying, like, what can I learn from you? What needs to be changed and what can be similar? RareX really brings forward the standardization domain area, meaning that collecting standardized data is really important, especially in rare disease, because towards clinical trial development and in engaging with clinical organizations, like we'll hear about from Michael, it's really important that we are collecting data in the same way. Though oftentimes we do need offshoots. We need things that are specific to our genotype, and that comes a little bit later. But making sure the foundation of our disease characterization and our disease data collection is in that standardized way. This really helps you to identify the therapeutic targets. What's out there? Is it new drug discoveries? Is it drugs that are out there for repurposing? And also the design of your clinical trials. In rare disease especially, we become partners with biopharmas that are coming to the space because we're the experts in our disorders. We're the experts to say, this really needs to be an outcome. This is how you can identify it. This is how you can see over the change over time. And if we're collecting data at the beginning and in a way that's really, really useful, our biopharma partners can utilize that to really design a clinical trial that's going to hit the mark. We see too many times that these trials are designed in ways that it doesn't hit the mark. And if through data collection from the beginning, being very, very careful, it's going to help us accelerate all of these approvals of these clinical trials and then drug marketing and approvals. It helps also determine outcome measures. Oftentimes the outcome measures that are out there, because we're rare, might not be ones that are really beneficial to the community, or what might not be ones that hit the mark again. We are often talking about biomarkers. We often talk about quality of life. We often talk about, in my spaces, we talk a lot about seizures. We talk about epilepsy and seizure number. Are those the best outcome measures for the drug target or what they're really looking at? Or do we need to have some experimental things or secondary outcome measures that can be tried throughout the process? And have other experimental therapies made a difference? Or anything else that there's interventions that your community is going through that you could say, oh, this seems to be something that's changing the trajectory of the disorder, or this is not, but it's oftentimes used. Then engaging with your patient and support with our quality care. at the end of the day, data collection and the characterization of your disorder really means a lot only when you bring it back to your patients. It really needs to be brought back into your community. The data you're collecting, how you're using it, what characterization is coming forward, how biopharma is using it, and it can be utilized by biopharma. And additionally, any physicians or medical professionals or researchers who are able to use that data is really, really important too, because you're going to improve your patient and community engagement, and you're going to find your best practices and best treatments through data collection. This is kind of the soup to nuts of what it really means to be a great, setting up a great organizational data collection structure and engagement. Because at the end of the day, the key to rare disease clinical research is to ensure that data collection meets the needs of all stakeholders and ensures that it really is truly patient-centric. And in order to do that, education around effective data collection, data sharing, and data usage is paramount. Because if we collect data and it's not able to be used by biopharma, and vice versa, if biopharma collects data, I'm going to put the stake in this ground. If biopharma collects data on rare disease patients or any patients at all, and it's not available back to the patient communities, and we're doing a disservice to both sides. So it's really pushing that forward. So happy to answer any questions at the end, but I'm really, really happy. I'm really, really proud to be able to turn it over to our other panelist. And I'm trying to advance my slide.
No problem. I'll just take a moment to introduce Michael to Vanessa. Thank you so much for sharing that. I personally have learned so much from you in the time that we've worked together. So thanks for that amazing presentation. So as Vanessa said, we're going to turn it over to Dr. Michael Grosso, who is the head of clinical development and specialty medicine at Daiichi Sankyo. And as global head of clinical development, specialty medicine, Michael's responsible for preclinical, clinical, phase 1 through 4, and the life cycle management of specialty medicine portfolio globally for, and get ready for this, orphan rare genetic disease, immunology, pain, neurology, inflammation, ophthalmology, cardiovascular, metabolic, and thrombosis, thrombolysis, hematosis, therapeutic areas. He is a board-certified academic cardiothoracic surgeon with 14 years of experience in the pharmaceutical industry. And Michael, I don't know how you have time to get all that done, but we're excited to hear from you.
I delegate a lot of work, but thank you, Kari. Thank you, Vanessa. Thank you to Global Genes for the opportunity to participate in the Rare Drug Development Symposium. I've attended in the past, and this is my first time for the opportunity to present, and I really appreciate it and respect the great work you all have done. As Kari mentioned, next slide, please, Vanessa. I work for Daiichi Sankyo. I've been there about 11 years now. It's a mid-sized pharmaceutical that started 100 years ago or so in Japan and merged into a global pharmaceutical back around 2007. But traditionally, the company in all those years was focused on cardiovascular metabolic disease, things like hypertension, diabetes, and atherosclerosis. But the leadership of the company was smart enough to recognize that with the growing and aging global population, there was going to be huge unmet medical needs in other areas. And they undertook lots of different research and surveys and recognized that the real unmet medical needs going forward would be in the areas of neurological diseases, cancer oncology, and in rare diseases. Next slide. And so with that, we made a switch from focusing on developing traditional oral small molecules to treat very widespread diseases like, as I mentioned, hypertension, diabetes, atherosclerosis, and recognize that to conquer diseases like cancer and rare diseases, we were going to have to make a big commitment to understanding new therapeutic modalities, things like antibodies and antibody drug conjugates, nucleic acid therapy, gene therapy, cell therapy, new drug delivery systems and vaccines and so forth. And with that, the company did make that commitment and focused our research laboratories many years ago to start this work. Next slide. And with that, the programs has finally started to mature to the point where we now have 3 programs in clinical trials treating patients to test our potential therapies in 3 rare diseases: Duchenne muscular dystrophy, pseudoxanthoma elasticum, and an ultra-rare disease, fibrodysplasia ossificans progressiva. But we also have programs that are just about ready to start in patients in other rare diseases like Netherton syndrome, glycogen storage disease, chronic inflammatory polyneuropathy, rare eye diseases, including retinitis pigmentosa, and several others. Next slide. But we also recognize that there were special strategies and tactics that would be important to establish a foundation for success if we were going to be able to bring treatments to patients with rare diseases. And our goal again was to develop world-class specialty and rare disease treatments and establish a clinical development capability for our what we call specialty medicine assets, utilizing these new technologies and modalities that I mentioned to you. We also recognize that we need to be able to quickly determine which one of these therapies have the most promise, and therefore we've put in a lot of mechanisms in our preclinical work to quickly demonstrate whether or not these molecules are actually working through mechanisms that we call proof of the mechanism or proof of concept. But we also recognize that there were important tactics that needed to be put in place that were different from traditional drug development. And first and foremost, we needed to recognize that we needed to fully leverage our strength in science, medicinal chemistry, manufacturing, clinical trial design, and execution. And I'm proud to say I've been with the company now for 11 years because of their, what I believe is their great commitment to science. We also recognize that we have knowledge gaps, that we're entering fields that we don't have experience in or have little expertise in. And with that, we need to either in-license or partner or collaborate to accelerate these technology gaps. And I'll share some examples of that in an upcoming slide. We also recognize that we need to interact quickly with the academic communities, the key opinion leaders, the key experts, the academic research organizations, in order to obtain natural history studies or access to registry data, and not through just these groups, but through groups, as Vanessa has pointed out, like RareX and other foundations, in order to be able to, again, rapidly gain as much knowledge as we can and be able to move these clinical development programs forward. Finally, we recognize the importance of working not just closely, but as real partners with the patient advocacy groups. And again, I'll give a couple of examples of that. And finally, utilize and leverage the expedited regulatory pathways that may exist for rare diseases. And what we found is that our foundation partners and patient advocacy groups know this better than we do. And again, having them as a partner is a fantastic benefit and experience. Next slide. So I did mention that one of the key foundations for success is recognizing where the knowledge and technology gaps are, and we certainly have done that. And with that, in order to bridge those gaps and accelerate the learnings, we've been very aggressive about forming collaborations with companies such as ERS Genomics in order to get our CRISPR gene editing therapies quickly up to the latest developments, and also companies like Ultragenyx, who have great experience and have already launched treatments in genetic diseases. Next slide. And as I mentioned, one of the most important foundations for success is not just collaborating, but truly partnering with the patient advocacy groups and foundations that represent these patients. First and foremost, they can be the primary source of clinical information on the disease, particularly patient databases and natural history where the medical literature itself may not have lots of information. They can communicate and negotiate with regulatory agencies, sometimes to even a greater degree than the pharmaceutical industries can. They've been involved with clinical trials, and they can help us design clinical trials and avoid all the some of the mistakes that have been made in the past. And most importantly, identify clinically meaningful endpoints, things that really will make a difference in the patient's lives that they represent. They also obviously help connect us with the patients and enhance enrollment for the clinical trials and spread the awareness of the diseases. And we've been fortunate so far to work with 3 great patient advocacy groups, Parent Project Muscular Dystrophy for our DMD programs, PXE International for our pseudoxanthoma program, and IFOPA for the rare disease fibrodysplasia ossificans progressiva. And I can tell you, I've been, as Kari pointed out, I've been in the industry about 14 years now, and one of the best experiences I've had, one of the most rewarding experiences I've had, one of the most collaborative experiences I've had has been working with these patient advocacy groups. And it's just been a tremendous experience working with these groups. And we're really looking forward to working with others as we move the rest of our research pipeline forward. Next slide. We're also trying to hold ourselves to a very high standard. We don't just want to walk around and pat ourselves on the back. And so we've put into place some very objective measures about how to determine whether we're succeeding or not. And we broke those down into 3 categories. First and foremost, the number of rare disease designations we get from regulatory agencies, things like breakthrough therapy, orphan disease designation, and in Japan, Sakigake designation. We're also going to measure the actual number of clinical trials that we really have underway. I mentioned we have 3 already. And finally, the number of new therapies we actually bring to patients. And I'm glad to say, even though we just started this venture just a few years ago, we already have 2 compounds that have received regulatory designation as either an orphan drug or Sakigake. And we hope to have another 2 programs underway in this fiscal year. So by the end of our fiscal year 2021, which would be March of 2022, we hope to have at least 5 clinical programs undergoing in 5 different orphan and rare diseases. And so with that, I'm happy to share some of the challenges that we faced in making this pivot from a traditional cardiovascular medicine company to a company that now is focused on orphan and rare diseases. But I can tell you would not have been possible without all of the help and collaboration and partnership from the foundations and patient advocacy groups that represent these patients. And so I'm happy to be part of this discussion today. Thank you.
Getting some questions from the audience, but I thought that I would start with one that I think both of you might be able to answer, which is, what are some of the most common pitfalls or roadblocks that you see come up in rare disease research? Who wants to take that one first?
I mean, from my perspective, it's recognizing that the traditional ways of doing things are not going to work. The standard drug development processes that have been so successful for the pharma industry in bringing new drugs to the market and to patients just don't hold true for rare diseases, in the sense that, number one, in many of these diseases, there's very limited numbers of patients. And so you can't do the trials that are designed as the way in the past, where it took thousands of patients whom you had access to in order to get a statistically relevant endpoint to prove that the drug or the therapy that you're testing actually work with clinical endpoints like how many patients actually, or how much longer they live, or changes in function of the heart and the lungs over time. What we've learned is in order to bring these drugs forward in the development with the very limited number of patients that are available to test it in, we have to use very different techniques and strategies. And we're starting to focus on a lot of different ways to measure success, looking at changes in biochemical markers or changes in imaging parameters, things that traditionally the regulatory agencies did kind of frowned upon as true indicators of actual treatment success. And that's where the patient advocacy groups, I think, and foundations have come into a great role, because they've actually educated the FDA and the other regulatory agencies to realize the benefit of these surrogate endpoints and the importance of these surrogate endpoints, and the realization that there's never going to be any treatments brought to the market and to these patients unless they start to accept these and build in some flexibility in their programs. And it was important that it didn't come from the pharmaceutical industry, because it kind of looked self-serving when we said it. But when it came from the patients and the patient advocacy groups and foundations, it made a huge impact. And I think there are numbers of examples of that already that we have seen. And I think that's one of the big differences that I've seen in working on programs that are focused on rare diseases as opposed to more traditional diseases. Vanessa?
Thank you. Yeah, I was going to say, Vanessa, from your viewpoint.
Yeah, I think that the big thing, Michael, you brought it up actually in your presentation, where how you partnered with the organizations, where you said the patient advocacy groups actually have the most up-to-date data about how the presentation of the disorder is happening. And that I think that happens with all of our groups, where we have publications out there, but they tend to always be, by the time they're published, 2 years down the road, they're defunct already. There's different things that we're seeing in the progression of the disorder, especially as the community gets older. So I think one of the biggest pitfalls that I see kind of with every group that I've ever worked with and that are coming onto RareX is when we go to collect data, everybody has the best of intentions when they start. And what ends up happening is, without kind of an eye on standardization and an eye on all the stakeholders involved, you tend to collect a fair amount of data that ends up getting a little bit noisy, a little bit messy, and not so useful for the end game. I think that all of us as rare disease patient groups, it ends up being that we want therapeutics. We also want to see what interventions work on our families and our kids. And to do that, really collecting data in a more standardized way is really important. So I think that has been the biggest pitfall. I mean, Kari, we've worked on data aggregation projects together in diseases that look very, very similar. And the way that we collect data became an issue, because we couldn't do cross-disease research because it wasn't a standardized way. So I think that's the biggest pitfall, is when you are told that rare disease groups go, okay, we're going to collect data, without kind of taking a step back and saying, okay, what data do we have available? What can we learn from it? If we have nothing, let's make sure that we start some standards and make sure stakeholders are all at the table at that first juncture.
And maybe Vanessa, I'm sorry.
I was going to say, I completely agree with Vanessa. On the first iteration of our natural history database, we collected everything under the sun and felt like it was going to collapse under the weight of all of it. So you're 100%.
And more data is not always better. That's the thing. The more data is always better. Or not always better.
And I think one thing, Vanessa, that I've learned, the pharma industry has been very protective of their data that they collect. And we're never going to make the progress that needs to be made unless we're all willing to share data. And the patient advocacy groups and the foundations have held the pharma industry's feet to the fire to share that data. And I think that's been a great thing.
Yeah, it has been.
That was one of the questions in the question box, which was, how can foundations encourage biopharma to share data and specimens they collect on rare disease patients?
I mean, it may be a little bit bold, but some of the foundations or groups that we've worked with have made it an upfront statement. Unless you're willing to share your data that you collect with all of the groups that we work with all of the industry people that we work with, at least to some degree, then you can't participate with us. And again, the patient advocacy groups and the foundations really are the drivers and the power brokers here. So don't be afraid to flex that power.
It's very true. And also I often talk when I give my larger Rare X talk is that a lot of it is the technology that exists for data sharing and data aggregation that existing today and didn't exist back, when rare disease was kind of getting its start. And I always say trusting the tech and now enabling better governance towards it, where they have the flexibility to share data in a way that can make everybody comfortable. Obviously, and when you're coming into IND, when an approval's on the line, it becomes a little bit more dicey. But there are ways to do it that weren't available back in the day and weren't an option. And I think really kind of coming into this century in that sharing and that collaboration is really important. And the patient groups requesting that and saying, we know this exists. We might not have it at a thing, but we know it exists. And how can we work together to do that?
Vanessa, this is like taking a little bit of deep dive because people are saying, we know you love data. So, but what kind of data? Where do these new starting out organizations collect it? And they need some direction for new rare diseases starting a foundation. So what is the data, what kind of data, and where do they collect it?
Okay, let's start first, but I'll address the foundation thing. But when your community is getting together, oftentimes we have amazing like Global Genes and NORD and all the other kind of helpers and to create foundations and organizations. At the end of the day, that takes time too, right? To create an organization, to create a foundation, to create a nonprofit, that is no longer a pitfall to inhibiting data collection. You can start data collection prior to having a community or an official organization. So we'll talk about the establishment of a foundation in a little bit. But in terms of the data collection aspect of it, there's a lot of efforts out there, including RareX, where it is that anybody can come onto the platform, even if they don't have an organization, even if they might be on their diagnostic odyssey, right? So they're not even sure what their diagnosis is, but they know they're in the rare categories. They're still going through their genotype, phenotype sort of thing, but they're wanting to say, hey, I'm here. I've got something I know is rare. I don't know what it is yet. I don't know where it's going to go, but I want to at least stand up and I want to be counted. So there's efforts like that through RareX. If you're a foundation that's interested in starting to collect data and working with biopharma partners, making sure to reach out to efforts that are out there and evaluating what you need versus oftentimes patient groups, they go into what they want and it's really, really big. And it almost is impossible to get your arms around, especially as you're starting. It's backing it up to say, what do you need? Can you govern it yourself? And then how do you put it into place? And also, can you budget it? Because it's not always cheap. I know Kari, do15Q, we spend a lot of money on our data collection efforts, but I think that that's the biggest thing, that taking a step back and identifying what you need, engaging your key opinion leaders in the space, whether it be academics, whether it be clinicians, the people that are kind of rising to the top. Rodney was just on the last panel. He's one of those in a lot of the rare disease space, especially in neuro, that he's a great key opinion leader to reach out to because they can network and find the people that can help you collect, set up your data structure. And additionally, always knowing that bringing it back to the patients, meeting their needs too. Meaning you're asking the questions that make sense to them and that's going to engage them in that data collection. And also letting them know when your data is being used. What are you going to do with it afterwards? How are you going to tell them how many people enrolled? What are the symptoms that are coming out? It doesn't need to be big papers. It doesn't need to go into the biggest journal that ever lived. It just needs to get out there in some space and get back to the people like Michael, where the biopharma partners are at least saying, hey, that phenotype looks really similar to this phenotype, which is in our pipeline now. So maybe this is a good conversation to start. And I think that's kind of when you look at that holistic view is, more data is not always better. That's the first case. Any question you ask should be able to be followed up later, but you don't need to ask all the follow-up questions from the beginning. Excellent,
Vanessa. And in the 2 minutes we have left, so not a lot of time, Michael, there was a question following up to Vanessa's. You have great partnerships with patient advocacy groups, but how should a new foundation seek out an organization such as yours to drive interest? As a follow-up to Vanessa's last statement.
That's a great question. There's probably really no formal process for that to happen. But I would say don't hesitate to pick up the phone and start to reach out to any name that you can find on the website on a particular pharma that seems to be interested in in any area that's even remotely close to what you're interested in, you'll probably have to reach out to a half a dozen people before you finally do reach someone who is either willing to talk to you or knows what you're talking about. But I think persistence, I think Vanessa and Kari, you can point to it, the success of the patient advocacy groups and foundations I've worked with all comes down to one thing, just persistence.
Excellent. Thank you both so much for being on today's panel and sharing such amazing information. I can't wait to knock on your door, Michael, and Vanessa can't wait to collaborate some more.
Yep.
I'd be happy. I'm looking forward to it.
Rodney, I'll turn it back to you for closing remarks.
Great. Thank you so much, Michael and Vanessa, for leading that great discussion. And Kari, I mean, this was just amazing. I was sitting here having my lunch and just taking notes furiously. Rare X, I think that is the bomb in the vernacular. And Michael, cannot wait to collaborate with you as well. So thank you to the audience for your questions. I would like to also thank Global Genes and co-host the Orphan Disease Center, Penn Medicine, for helping make this happen. So I would like to say for closing remarks, in addition to thanking all the speakers of our panel and discussion leaders for today, would like to thank our symposium sponsors, including Greenwich Biosciences, Horizon, Pfizer, Applied Therapeutics, Catalyst, Daiichi Sankyo, Harmony, Ionis, Janssen and Johnson, Passage Bio, Farming, Regeneron, Spark, Tisha, Ultragenics, Amicus, Avidity, Check Rare, Forge, Genentech, Ovid, and Regenix Bio. Thank you all for your support. And next, please join us for the upcoming office hours. Oops, next here for one of the two office hours, as we've been talking about these office hours earlier and throughout the entire day. Join us, whether it be for the preclinical or clinical discussions, to really continue this chat about the journey, how to work with foundations, how to work with academics, how to work amongst all of these people, and how to find those connectors who will help enable you move your portfolio or move your foundation forward. So thank you. And finally, please take a few moments to respond to the polls that are being launched now. We all appreciate your feedback on the sessions and want to ensure that we are consistently advancing our programming in accordance with the needs of the community. And you could also share your feedback via events@globalgenes.org. And one last note, take some time, if you can, to explore the event page, because the next 2 days is going to be wonderful, excellent, fantastic, you name it, just great lineup of speakers and content. Visit the posters, build your itinerary for the remainder of the week, and plus visit the Achievements and Badges tab to learn more about how you could win a pass to a future Global Genes program, which would be really neat if we could be in person again one day. So with that, thank you again. And I will sign off and go into the preclinical foundations room. And Kari, do you have any last words?
Well, I'll be in the clinical research room, so I look forward to having you all there with us. And thanks again, everybody, for participating today. The chat has been really robust, and I hope you've made new connections, and really appreciate the thoughtful questions that people asked during our panel discussions. Thank you all so much.
Thank you.